Bom dia, everyone. Good afternoon from Belgium, or good morning in Brazil. And I would like to thank the Brazilian Journal of Analytical Chemistry to provide me with the opportunity to discuss some of the work we carry out at Ghent University. And I would like to focus on the use of ICP-mass spectrometry in the biomedical sciences and try to answer the question whether this is a workhorse or a racehorse.
Thank you.
First of all of the work that I will present is, of course, done by my team at Ghent University. And this is a recent picture that dates back to our team building activity some weeks ago. For those of you that are not that familiar with ICP-mass spectrometry, this is in fact the most powerful technique for trace and ultra-trace element analysis, and it's typically used for the analysis of liquid samples, aqueous solutions, and acid digest of solid materials. And the ion source is actually the plasma, the inductively coupled plasma, which can be considered as a hot electrical flame that has a temperature that is so high that all the molecules are actually broken down into atoms that are subsequently ionized. And most of the ICP-MS instruments still today are equipped with a quadrupole filter for mass analysis. To convince you that this is a very powerful tool for elemental analysis, I would like to show some detection limits. And you can see that for the majority of elements, metals, we can obtain limits of detection below 1 nanogram per liter. And for those elements that either suffer from spectral overlap and or have a high ionization energy, limits of detection are slightly deteriorated, but still at trace and ultra-trace level. ICP-mass spectrometry was commercially introduced in 1983, and then the instruments looked like this. So they were very large, and they had still a number of buttons to tune the instrument and so on. Currently, we have different types of ICP-mass spectrometers, different sizes, different colors, as you can see. But more importantly, we also have ICP-MS instrumentation that is equipped with different types of mass spectrometers. Still, the majority of instruments is equipped with a quadrupole filter for mass analysis, but there are also instruments that are equipped with a double focusing sector field mass spectrometer, so consisting of a magnetic and an electrostatic sector, either in near-Johnson geometry or reverse near-Johnson geometry, and equipped with a single detector or with an array of detectors in a multi-collector setup. There are also tandem ICP-MS instruments that are equipped with a collision reaction cell that is located in between two quadrupole filters and that provides a nice approach to tackle spectral interference. And then there is also instrumentation that is equipped with a time-of-flight analyzer for mass analysis. And with this type of instrumentation, for a package of ions, we can obtain a full elemental mass spectrum. Biomedical applications is also nothing new. As of the late 1980s, papers started to appear in which the determination of trace and ultra-trace elements in biofluids like whole blood or blood serum, urine, and so on were starting to appear. And ICP-MS is nowadays used in many clinical labs as a routine tool for the determination of either toxic or essential mineral elements. But I would like to focus on a number of more high-end applications, and I would like to start with elemental mapping using laser ablation, ICP-MS, which is sometimes also referred to as elemental bioimaging. In such an approach, a thin section of tissue is mounted in an airtight ablation cell. And by means of a video monitoring system and an objective, we focus a high-energy UV laser beam on top of the sample. And whenever we fire the laser, a small amount of material is converted into a dry aerosol and can be taken using a helium carrier gas into the ICP for elemental analysis. And what we can do is actually to scan the laser line per line over the tissue in order to reveal elemental distributions in 2 dimensions, and as I will show, sometimes even in 3 dimensions. Does this provide additional information? Well, I would like to answer this question by means of an example. And in this example, we focus on the peritoneum, and that is a membrane that lines the abdominal cavity, as you can see here, and it is inside all of us. And unfortunately, in some individuals, peritoneal carcinomatosis can occur as a result of metastasis of other cancers in the gastrointestinal or gynecological region. And these patients often show short survival and have a poor quality of life. And the treatment, which is palliative, consists of surgical removal of the tumors. And during the surgery, the patients are also treated intraperitoneally, so locally on the peritoneum, with oxaliplatin as a platinum-containing chemotherapeutic agent. And the oncologists are interested in assessing how effective the drug penetrates the tumor tissue. How is this normally done? Well, a small part of tissue is taken, this is digested, and then analyzed using traditional ICP-MS, so using solution ICP-mass spectrometry. This is how an H&E stained section looks like. So this is investigated by a specialist who indicated that what is indicated here are tumor tissue, so is tumor tissue. And here, for an adjacent section of the tissue, you have the platinum signal intensity with the yellow color indicating the presence of higher concentrations of platinum and the darker colors indicating lower concentrations of platinum. And what you can see, unfortunately, is that where there is tumor tissue, there is not a lot of platinum. So this was not good news because the drug doesn't seem to enter the tumor tissue very efficiently or effectively, but you cannot deny that this provides added value compared to, let's say, a general picture that only gives the average platinum concentration. So we started about 15 years ago. We did this type of elemental mapping work. And the first paper that we published on the topic appeared in 2010 and concerned this fellow here. So this is a water flea or a Daphnia magna. And this is often used as a model species by our colleagues from the ecotoxicology lab. And they had seen that exposure of this animal to higher concentration of zinc led to reproductive toxicity, so fewer young animals. So we looked at the element distribution in sagittal and dorsoventral cross-sections of the animal. And for instance, you can see phosphorus here, which indicates biological activity, calcium, the exoskeleton of the animal, and here zinc, which seems to be enriched in the embryos, and we could link that with the reproductive toxicity. This is a paper from 2012, so that in which we used laser ablation ICP-MS to map the presence of a bromine-containing drug, so the drug that you can see here, and its metabolites across the organs of a rat. That was of rats that were sacrificed at different time points after administration of the drug. Well, now I'm a bit ashamed to show these maps because they are not state of art, but in 2012, such an approach was considered as a hot paper, so a hot topic, and it featured the cover of the Journal of Analytical Atomic Spectrometry at the time. This was the unit that was used at the time, so it was a so-called New Wave Research Universal Platform 193H energy laser, and it was equipped with a supercell that you can see here. So a kind of teardrop cell, and this cell was developed such that the geometry and the flow characteristics of the cell were specifically designed to enable rapid evacuation of the laser-generated aerosol. And this meant that transient signal evacuation was complete in less than 2 seconds. Well, from the point of view of now, this is incredibly slow. and we don't like slow. Why? Well, first of all, this leads to long acquisition times. Secondly, if the signal is stretched over time, this deteriorates the signal-to-noise ratio. And as a result of this deteriorated signal-to-noise ratio, we are also obliged to use laser beams with a wider diameter, so some tenths of micrometer. In 2014, we got a new unit that was equipped with another type of cell, a so-called two-volume cell, which features a larger cell to house the sample, and then a smaller cell that hovers over the surface of the sample, and that led to a substantial improvement. But still, we were not yet at the situation that we wanted to be, and that is why we developed our own cell. Before discussing the cell, I would like to indicate how fast is defined, or how the speed of aerosol evacuation is defined. Well, for that purpose, we look at the so-called single pulse response. So this is the transient signal that we observe after a single laser shot. And then we measure the width of the curve, either at half of the intensity, 10% of the intensity, or 1% of the intensity. Actually, you can select, but of course, it's necessary that apples are not compared to oranges in this context. So this is the type of cell that was designed at the Ghent lab. And we have the sample here. The UV laser beam is focused through a window onto the sample surface. And the aerosol that is formed upon laser sample impact is transported away very rapidly from the point of interaction using a fast flow of helium that is flowing in a laminar regime. And we have patented this design and also licensed it to Teledyne photon machines, who, based on this design, came up with a cobalt cell that is now commercially available. We have also developed a so-called aerosol rapid introduction system, or ARIS. This is the transfer lane connecting this ultra-fast cell with the ICP torch. And this is characterized by a minimum dead volume and preservation of the laminar character of the helium gas that is transporting the aerosol into the ICP. Where are we at now? Well, this is what we see nowadays as a single pulse response. And you can see that the duration of a pulse, for instance, when ablating glass, equals 0.3 milliseconds when measured at half of the maximum, and half a millisecond, for instance, when measured at 10% of the maximum height. So this means that over the years, we have been able to reduce the pulse duration from a second to less than a millisecond. And this means that nowadays we can obtain two-dimensional maps at the pixel acquisition rate up to 1000 pixels per second. As a result of all the material being kept in a small plug, we also have a much better signal-to-noise ratio, and this allows us to use laser beams with a smaller diameter, for instance, down to 1 micrometer or even slightly below that in some occasions. As a bonus, we also saw that the absolute sensitivity increased, and that is because we avoid losses in the transfer line to the largest possible extent. Here you can see that we can have such shots one after the other, and that they are all shorter than 1 millisecond. And what is shown here is that actually the duration of a single pulse response is not affected by the laser repetition rate used, up to 1000 Hertz, and that also the integrated intensity is not affected over the entire range of laser repetition rates that we can use. This is where we are today. So what you see here is the distribution of copper and the distribution of zinc across liver tissue of mouse after common bile duct ligation. So copper is an essential element. We need it for our health, for our well-being, but the body cannot upregulate or downregulate the uptake of copper. So what the body actually does is, if there is excess of copper, it excretes this copper via the bile, and then it ends up in the feces. But as a result of the bile duct ligation, so cutting the bile duct, this exit was closed, and you can see that this leads to copper excess, to accumulation of copper in the liver under the form of hotspots. And the size here of such a map is quite large. It's 7 by 1 millimeter, more or less. It was mapped at 1000 pixels per second with a 5 micrometer beam diameter, and this only took us 17 minutes. Zinc remains homogeneously distributed under these conditions, as you can see. How do we have to present the samples? Well, we have thin sections that are mounted on a microscope glass, and we need to select the laser energy density such that there is hard ablation of the biological tissue, while the glass does not contribute to the signal, so that because the laser energy density is below the ablation threshold of the glass. And we have tested a number of brands and came to the conclusion that it's actually not well, we don't have to select a particular brand, because you can see that, for instance, at the laser energy density below half a joule per square centimeter, we already efficiently ablate biological material, while glass only starts to be ablated at higher laser energy densities. How do the craters look like under these circumstances? Well, we shot some craters in gelatine as a proxy for biological material, and then studied the shape of the crater using atomic force microscopy. And you can see that we have fairly straight walls and a more or less flat bottom, as you can also see here. We are not only interested in revealing the element distribution as such, but we also want to have quantitative information. And with laser ablation ICP-MS, this actually means that you need a matrix-matched standard that shows the same ablation behavior as the samples. And we typically select gelatine. Gelatine has a number of advantages. First of all, it consists of partially hydrolyzed collagen. And that's interesting because collagen is the most abundant protein in mammals. So up to 25 to 35% of the whole body protein content is collagen. Also, gelatin during the preparation at one stage is liquid, such that it is quite easy to add aqueous standard solutions. Then the gelatin is dried, and under oven drying, so under controlled conditions, we can have a homogeneous distribution of most of the elements in the gelatin. Gelatin also is effectively ablated, as it shows a maximum absorption around 190 nanometer, which is close to the 193 nanometer of the laser radiation. What can we do with the setup? Well, we can, for instance, have a look at metal-based drugs, at contrast agents, halogen-containing drugs, metallic nanoparticles, endogenous elements, and metal-labeled antibodies. I will show a couple of examples, but I would like to stress that these have been obtained over many years, so that means that they do not all have the quality of our most recent work. In a first application, we worked together with Johnson & Johnson, and they wanted to evaluate the distribution of platinum in kidney tissue of cynomolgus. And the platinum, once again, was coming from a chemotherapeutic drug, so a drug against cancer, and in this case, it was cisplatin. At first, we looked at a larger section of kidney, and we could establish that the platinum was predominantly present in the cortex of the kidney tissue. And then we selected a smaller region of interest and mapped the distribution of copper with a 1 micrometer beam diameter. Here you see the result for two adjacent sections. This section was H&E stained, so as is done for histology evaluation, and this is the laser ablation ICP-MS map for platinum. I hope that you can see that the circular structures that we see here also appear in the platinum map. And this actually shows that platinum is present in cortical tubular epithelium. Then we also have seen some hotspots for platinum, and these hotspots corresponded to necrotic tubules with slowed epithelial cells, so epithelial cells that have lost their connection and are present in the lumen. We can also have a look at the distribution of proteins. And this may sound a bit strange at first because normally ICP-MS is not suited to have a look at proteins. So what we need to do is to make these proteins visible or accessible using ICP-mass spectrometry. And this can be done using antibodies that are tagged with a metal label. So in the context of cancer studies, we, for instance, had a look at the distribution of the chemokine receptor 4 protein. And the expression of this protein is directly related to the malignancy and the metastatic nature of the cancer. There is a peptide that binds specifically to this protein. And at the Leiden University Medical Center in the Netherlands, they could label this peptide with holmium as a metal. So this means that if we look at the distribution of holmium, for instance, in liver tissue, that this corresponds to the distribution of this protein. And you can also see that in the presence of tumor tissue, we can also visualize the expression of this protein. We can also have a look at endogenous elements. This means elements that are naturally present in the tissue that we investigate. And here we looked at the intestine villi of mouse. So intestine villi are the small parts inside the intestine, so on the intestine's inner surface. And they serve to increase the surface dramatically in order to have an efficient uptake of compounds from the food. And here you can see a map for zinc. The distribution of zinc was mapped with a 0.6 micrometer laser beam diameter. So this is the smallest beam diameter that we have ever used. And this distribution or this area is about 300 micrometers by 200 micrometers and was mapped at 50 pixels per second. And what you can see here is actually that the zinc map is so detailed that we could see all of the structural micro parts, the microstructure of these villis, as you can see here. To some extent, we can also do three-dimensional bioimaging. And the first time we did this was for studying the metal distribution and metalloid distribution in single grains of wheat and rye. What we did is actually combine a number of 2D maps. What we did is we removed a bit of material using grinding paper and then obtained a 2D map. Then again, we removed a bit of material, obtained another 2D map, and so on and so forth. And then all of these 2D maps were combined in order to reconstruct the three-dimensional distribution of elements like manganese, for instance, copper, and zinc across these individual grains. We have also done 3D mapping of elements in Cereodaphnia dubia. Again, this is a water flea, but a smaller kind of water flea than the Cereodaphnia magna that I've shown before. And here, in order to obtain a 3D distribution, we sliced the animal in a number of sections, and we mapped the distribution of elements, so in 2 dimensions, in each of these sections. Here it was more challenging to combine all of these 2D maps in such a way that we had the accurate representation of the distribution of elements in 3 dimensions. And in order to help us with that, we measured using CT scanning. We measured the animal before, and CT scanning gives us a 3D image of the animal based on differences in density, and the technique is based on the use of X-rays. And in order to further enhance the differences in density, we have exposed the body of the animal to uranium, and uranium is taken up in the body in a non-selective or non-specific way. And here on the left side, you can actually see the 3D image of the body of the water flea as obtained using differences by exploiting differences in the density using CT scanning. And on the right side, you can see the uranium 3D map. So with all of the 2D maps stacked such that we have a correspondence between the two 3D images. And that then also allows to have a look at other elements, such as here, zinc, nickel, and copper. And once again, you see that zinc, which is indicated in red, is present here in the embryos, which provides the link to the reproductive toxicity I've mentioned before. The best type of ICP-MS instrument to use in the context of laser ablation ICP-MS may well be an instrument equipped with a time-of-flight analyzer. And with a time-of-flight analyzer, if you introduce a package of ions, these ions flow or move with a different velocity through a tube, the field-free fly tube, and they reach the detector at a different moment in time. So this means that when you think about laser ablation, for every pixel on the map, we can actually have the full elemental mass spectrum. And this is what was done here for the study of the metalome in embryos of mice. And I've selected a number of elements here, but in fact, we get similar maps for all of the elements in the periodic table. And here, the acquisition rate was 250 pixels per second, and the spot size used was 10 micrometer. I would then like to switch to another topic, and this is the quantitative analysis of single cells, so the determination of metal contents in individual cells. And perhaps some of you have already heard about single particle ICP-MS. And this is an approach whereby ICP-MS is used to characterize nanoparticles, typically engineered nanoparticles, which have a size below 100 nanometer. For this application, we introduce a dilute suspension of nanoparticles into the ICP. And whenever such a nanoparticle is entering the ICP, we get a burst of ions, and that can be measured as a short transient signal, again, with a duration of about half a millisecond. Here you can see the raw data. So for every of these nanoparticles reaching the plasma, we have a transient signal. Then, as a first step, the frequency with which a given signal intensity is observed is plotted as that signal, as a function of that signal intensity. And then we get this type of histogram where we have the background signal here, which is due to analyte element in solution, dissolved in the solution. And here we have the part of the histogram corresponding to the nanoparticles. And upon proper calibration, this can be converted into a size distribution. Well, single cell ICP-MS works in exactly the same way, at least concerning the basic operating principles. But a cell is quite different in size than a nanoparticle. Per definition, a nanoparticle is smaller than 100 nanometer, while a cell, depending on the cell type and whether it comes from a plant or an animal, for instance, can have a size between 2 and 100 micrometer. So to put this in perspective, the blue circle here represents a water droplet with a diameter of 4 micrometer. And the small yellow dots here are then nanoparticles with a diameter of 50 or 100 nanometer. And here we can use the standard sample introduction system, but this is not the case for cells. So this red circle here represents a cell with a diameter of 50 micrometer. And actually the blue circles are water droplets with a size of 4 micrometers. So we are in a very different size regime. And as a result of this, we can actually not use the standard sample introduction system anymore, because this is focusing on reducing the size of the droplets introduced into the ICP to 10 micrometer or lower. But by now, commercially available sample introduction systems are available that introduce cells into the ICP with a transport efficiency of some tenths of %. Another challenge is that cells are much more fragile. And that's why sometimes, or most of the times, I would say, fixation is required using paraformaldehyde or glutaraldehyde. What is actually done is protein chains are cross-linked, and this leads to entities that are more rigid, that are more rugged, and can be introduced as such into the ICP. Now we have done an application, carried out an application, whereby we had a look at 3 types of human cancer cells. And these cells were exposed to cisplatin as a chemotherapeutic drug at different concentrations. And then we were looking at the uptake of the drug in the different types of cells. And we also evaluated potential differences in essential elements like phosphorus, sulfur, copper, iron, and zinc. What is the added value here? Well, of course, if we take a larger sample of cells and we digest the cells, we can determine the average concentration of, for instance, the drug in the cell population. But if we look at the individual cells, we can distinguish between these situations. So do all cells have all cells taken up equal quantities of cisplatin? Have only a few of the cells taken up larger amounts of platinum? Or is there a wide variety of the amount of platinum in the cells? This is what we studied, and you see some raw data here, for instance, for Raji cells exposed to 0, 5, 10, 20 micromolar of cisplatin. And here you see for one type of cell, so the same type of cells, the histogram for the control sample and those after exposure to 5, 10, and 20 micromolar of cisplatin. You can see that the average or median concentration that has increased, which is of course no surprise, I would say, but it's also clear that now you have information on the distribution of the platinum amount in the cells. We can also look at those histograms for the other elements, so like phosphorus, sulfur, iron, copper, and zinc. And we can also calibrate these signal intensities such that we have information on the absolute amount of metals in these cells. And you can see that for the metals, this is at the femtogram cell per cell level. And for copper, we can even go below 1 femtogram. What was interesting to see for us is that one of these cell types absorbed more of the drug than the other 2 cell lines. And this could be linked actually with the effect such a drug has on the well-being of the cell. You can see that the cell viability for this type of cells, Jurkat cells, is reduced much more efficiently than for the other types of cells. And this could be linked with a higher uptake of platinum. Back to laser ablation now, because we can also use laser ablation for quantifying the cellular uptake of a drug. So to determine how much of the drug has entered the cells. And this was done with human breast adenocarcinoma cells, which were exposed to a candidate drug against cancer based on copper. And so what we did is after exposure, we mapped the cells. So here you can see a microscopic image of the cells. And here you can see the copper map. And I think it's clear that copper has indeed entered the cells, that the cells have taken up copper. And you can even see that copper is predominantly present in the nuclei, where it's supposed to bind to the DNA, so where it's supposed to be active. Also here, we used gelatin droplets as standards with a known amount, absolute amount of copper. And upon ablation of these droplets, we could see that the integrated intensity varies linearly as a function of the absolute mass of copper in such a gelatin droplet. And the limit of detection was 10 attograms of copper. And here you can see in blue the copper amount, 5 femtograms, in the control cells. There was already some copper there because it's an essential element, but you can also see that the copper concentration has increased in the cells after incubation with the candidate drug. Now, for the last section, I would like to have a look at the isotopic composition of essential mineral elements in the context of biomedical applications. And this is based on the observation that all elements with 2 or more isotopes actually show natural variation in their isotopic composition. And this is the result of isotope fractionation. This term refers to the fact that isotopes of the same element may take part in physical processes, chemical reactions, biochemical reactions with a slightly different efficiency. And this different efficiency can be the result of both small differences in the reaction rate, so in kinetics, or in the equilibrium state, so thermodynamics. A couple of rules of thumb. The lighter of 2 isotopes always reacts slightly faster. The heavier of 2 isotopes has a slight preference for the strongest or hardest bonding environment. Kinetic and thermodynamic fractionation can work in the same direction, but they can also counteract one another, so it's not predictable or not always predictable. And isotope fractionation is larger for the light elements, because then the isotopes show a larger relative difference in mass, and also tends to be a bit larger for elements that occur in nature in different oxidation states. In any case, we are talking about very small differences in isotopic composition. And that is why we need a special type of ICP-mass spectrometer for this type of applications, and this is a multi-collector ICP-mass spectrometer. So this unit is equipped with a double focusing sector field mass spectrometer with an electrostatic sector and a magnetic sector, and very importantly, an array of detectors, typically Faraday cups, and these allow the simultaneous monitoring of the intensities of several ion beams. And that's actually the key to very good isotope ratio precision, which can be as low as 0.001% RSD under ideal circumstances. That's the measurement precision. As we are dealing with very small variation, very small differences, the results are typically expressed as delta values. And this is the relative difference between an isotope ratio in a sample and the same isotope ratio in a standard, preferably an internationally accepted reference material. And then this difference is multiplied with 1,000 in order to express the result in per mil. So we are talking about delta values expressed in per mil. Let us first have a look at the copper isotope ratio and its potential use as a diagnostic or prognostic marker in liver disease. And I've told you before that copper is an essential element. It is taken up from the diet in our intestines and is then bound to a chaperone protein, because as an ion it would harm our tissues. So it is bound to a chaperone protein to be transported to the liver. In the liver, the copper is incorporated into ceruloplasmin and is re-released into the blood to be transported to where it is actually needed. The body cannot upregulate or downregulate the copper intake, but excess of copper can be removed in a healthy individual via the bile and then the feces. As a result of the central role of copper in the copper homeostasis, in the copper metabolism, we looked at copper isotope ratios and copper concentration in blood plasma of patients suffering from liver cirrhosis, which is a serious liver disorder. On this axis, you see the copper concentration, and data for liver cirrhosis patients have been indicated in red here, red data points, and for a reference population or a control population in green. First of all, when you look at the concentration, you can see that there is not a lot to observe because the ranges overlap completely. But if you look at this axis, this represents the delta copper value, so the copper isotope ratio, and you see that for patients there is a systematic shift towards a lighter copper isotopic composition. Here, the copper isotopic composition has been plotted as a function of the melt sodium score. And what is the melt sodium score? This is a prognostic score that is used in hospitals, and it's kind of an estimation what the chance of a patient is for surviving a given time window. Well, this does not sound very, very nice, but actually it is done in order to prioritize patients for liver transplantations. There are always fewer donor livers than patients requiring liver transplantations, and those patients that have the worst condition, that needs a transplant liver the most, are prioritized. And you can see that the worse the condition of a patient is, the lighter the isotopic composition typically is. In end-stage liver disease, the only therapy that is left is the liver transplantation. And we had a group of patients, 32 patients, that were followed before and after liver transplantation. So they are another group of patients than the liver cirrhosis patients. Here we had end-stage liver disorders of various types, various etiologies. But you can see that also in this patient group, we have delta copper values that are lighter than those for the reference range, which is here. But already 3 months after the liver transplantation, we see some normalization. Data points are shifting in the direction of the reference range. And this was even more the case 9 months after the transplantation, at least on condition that the patient was doing well. Because here, patient 31, for instance, that was not a very nice story. So at first, we did not see a lot of variation in the delta copper value. But then all of a sudden, the delta copper value dropped dramatically. And unfortunately, this patient was diagnosed with recurring liver cancer with metastasis in the lungs and the heart, and died some months later. So this seems to indicate that following up on the copper isotopic composition in blood serum provides information on the well-being of the patient. As a last application, we also had a look at isotopic analysis in the context of Alzheimer's disease. And of course, you know that this is the most prevalent form of dementia. And it is characterized by the formation of 2 types of protein aggregations in the brain. And these protein aggregations consist of beta amyloid plaques or neurofibrillary tangles of tau proteins. And it is believed that some metals play a role in the formation of these protein aggregations. And one of the suspect metals is copper. So we used a mouse model for Alzheimer's disease. These are genetically modified mice, which shows all of the symptoms of Alzheimer's disease. And we compared the values for these mice to the corresponding wild-type mice, so which are not genetically modified. And in order to separate development of the disease with aging, we also had a look at an old population and a young population of mice. And again, we measured the copper 65, copper 63 isotope ratio in blood serum and in several individual parts of the brain, like brainstem, cerebellum, cortex, hippocampus, and so on. Here on the left, we had a look at copper concentrations. And the Y stands for young, so we have healthy young individuals, mice, and young individuals with Alzheimer's disease, genetically modified, and the same for an old population, healthy and diseased individuals. And in terms of concentrations, we can say that aging seems to increase the copper concentration in blood serum, but the development of Alzheimer's disease does not lead to a systematic change in the concentration. Here we look at the copper isotope ratio in the blood serum, and you see a more systematic change towards a heavier isotopic composition or towards lower, or let's say more close to 0 delta copper values that are closer to 0. Next, let's have a look at the cerebellum. So this is this part of the brain, and it's the location of muscle control, including balance and movement, and also some cognitive functions are located here, including the language processing and the memory. This is the concentration in this tissue, and again, we see that it's not that clear. We could say that to some extent aging leads to a higher copper concentration, but the difference is not that clear. There is quite a bit of variety here. But when we look at the copper isotope ratio, we can see a more systematic shift once again towards a heavier copper isotopic composition. When we then plot the copper isotope ratio in the cerebellum as a function of that in the plasma, in this pilot study, we saw that the groups could be more or less nicely separated from one another. And this seems to indicate that aging leads to a lighter isotopic composition, and the development of Alzheimer's disease leads to a heavier isotopic composition. These are two examples only, but isotopic analysis of so-called essential mineral elements is an area of attention within our group, because very often, like in these cases, we see that isotope ratios are either more sensitive in picking up changes in the metal metabolism, or the isotope ratios may even contain, information that is different from that is present in the corresponding concentrations. So the question, is it a workhorse or is it a racehorse? One could actually say that ICP-MS nowadays is an entire stable of horses, and that the use of the horse depends actually on the analytical challenge, I would say. Before ending, I would like to thank a number of colleagues from different backgrounds, so biology, biochemistry, medical backgrounds, and so on, because without them, this type of interdisciplinary work would not be possible. I would also like to thank the funding organization over here, FWO, and some instrument manufacturers that have supported us. And last but not least, I would of course like to thank the Brazilian Journal of Analytical Chemistry and all of you for your attention and your participation this afternoon. So thank you, obrigado.